invitation to speak. And when I came to prepare this talk, I suddenly realized I haven't given a conference talk for over a year, I don't think, about my research. And many of you would have heard me speak in the past about the work my lab's doing on ash in particular. So I thought what I'd do today is just try and give an update on where we're up to with ash and also where we're up to on oak, because we're increasingly working on oak as well. And as Rob mentioned, we're part of the Action Oak group. So to start with ash, last year we published this paper in Nature Ecology and Evolution, Genomic Basis of European Ash Tree Resistance to Ash Dieback Fungus. And I'm going to tell you about some work we've done that we're doing that's following up on this study in a natural population. But before I do that, I'll just recap what we did, because it'll help when we start thinking about how our new data reflect on this previous study. So in this previous study, we were working on the forest research mass screening trials that were set up with DEFRA funding quite soon after ash dieback struck the UK. There's some massive trials. We chose the 4 sites where ash dieback damage was worst, but tree survival from establishment was the best. And we used this to do a genome-wide association study and to do some genomic prediction. And I'm sorry if you can hear a vehicle reversing outside my office. There's nothing I can do about that, sorry. For our GWAS, we used pools of DNA samples, and the pools came from trees with the same health status under ash dieback, but being from the same provenance, the same native seed zone. And this meant that we could look at allele frequencies within populations quite cheaply, this pooling strategy. And that enabled us to do a GWAS. And here's a Manhattan plot from the GWAS. So we found some sites within the ash genome that seemed to be quite significantly associated with remaining healthy under ash dieback. So we were quite encouraged by this. And some of the sites that were highlighted by the GWAS seemed to be the kind of genes that we would expect to be involved in response to a fungal pathogen. So that was all quite encouraging. And then we looked at 150 individuals that we did the whole genome sequences of individually. And we used our GWAS results and our allele frequencies from our pools to come up with a genomic prediction model based on thousands of SNP loci within the genome, so that we could try to predict what the health should have been of these 150 individuals. And then we could compare that to how healthy they actually were. And we got some quite encouraging results. So this shows a chart of our correlations between our genomically estimated breeding values, our estimate of how healthy these trees should be, and therefore how useful for future breeding, and their actual tree health, their actual health under ash dieback. And you can see here that with many thousand SNPs, we could end up with breeding values that correlated reasonably well with actual tree health. So we're getting correlations of between 0.3 and 0.4 when we're using over 25% of SNPs to do the calculation, to train the model and do the calculation. And for genomic prediction, this is quite strong correlations. Genomic prediction is often used in breeding of things like cows for milk production and beef production, or pigs, or and increasingly in plant crops as well. And these are the kind of correlations that are good enough for breeding programs. So we're quite encouraged by all this. So what we thought we would do next is go out into a natural woodland and look for what's happening there regarding tree health and the frequencies of different alleles within the genome. So we looked quite carefully for a suitable woodland. We wanted a woodland where there were adult trees that were affected by ash dieback, but also a lot of natural regeneration going on, so that we could look at the new generation of trees that were coming up. And we eventually selected this wood, which is Marden Park Wood in Surrey, which is owned and managed by the Woodland Trust, and they've been extremely helpful to us in this study. There's lots of ash dieback here. When we first went in, it was early autumn, and you could see the fruiting bodies of the fungus abundantly on the ground there in the wood. As you can see from this drone footage, there are a lot of dead adult trees there, or at least adult trees with no leaves on. But you can see a few leaf canopies of ash trees there that are looking reasonably healthy even now. And on the ground, as I say, there's a lot of natural regeneration going on. And this is a on an ancient woodland site. It's in an area of outstanding natural beauty. It's a site of special scientific interest. And the Woodland Trust management plan going forward is not to be felling trees unless they actually pose a health risk to people walking on the paths in the wood. And the management plan is very much just to stop invasives from coming into the wood and just try and keep things as natural as possible. So we hope we can do a long-term study here. So in 2019, we went in, and in this patch of woodland, we sampled all of the ash trees and estimated their size and their ash dieback status. And we had three hypotheses, really. The first hypothesis is that frequencies of alleles at some loci will consistently co-vary with level of ash dieback damage in this woodland. Our second hypothesis is that many of these loci will be the same as we found in our previous study, based on the forest research mass screening trials. And thirdly, we thought the smaller younger trees might contain a biased subset of the variation present in the adult trees due to more rapid selection among them by Hymenoscyphus fraxineus, by the ash dieback fungus. So there's been quite a lot of studies that suggest that small trees die more quickly from ash dieback than large trees. So we were hoping that in this multi-generational population of ash, we might detect evidence in the genomes of the trees for natural selection happening in real time. And of course, this has policy implications, because if we discover that natural selection really is happening very fast in natural woodlands, then if nature just takes its course under the ash dieback pandemic, then we will very quickly see trees being bred naturally by natural selection in the wild for increased resistance. However, if we don't see that natural selection happening fast in the wild, it strengthens the case for a breeding program. We might need to accelerate the process. So this is a very interesting study, both in terms of evolutionary genetics and ecology, and also in terms of policy for what we do in the UK about ash dieback going forward. So here's the sizes of the trees that we collected data from. We've got their size here as an estimated diameter at breast height. As you can see, we've got a lot more small trees than we have big trees, and that simply reflects the actual frequencies of these different age classes in the woodland itself. It's a really nice wood for natural regeneration. There's not too many deer in this wood. In that way, it's quite unusual for England. For the juvenile trees, we scored ash dieback damage using a modified version of scoring systems that other people have used and that we used in our previous study on the forest research mass screening trial. So a higher number means a more healthy tree, and a 1 means a dead tree. But for the large trees, we couldn't use this system, so we just tried to estimate the leaf canopy cover as a percentage, 100% being a healthy tree and 0% being a tree with no leaves at all. And unfortunately, we just had to do that by eyeballing from the ground. We're hoping that we'll be able to do it with drone footage in future, and that's partly why we've got that drone footage that I showed you earlier, but we haven't yet been able to do anything more rigorous than just eyeballing from the ground. And a lot of this field work was done by members of my group, Laura Kelly, Jonathan Stocks, Kerry Metheringham, and Will Plum. Here's the health of the trees in 2019. The juvenile trees have a lot of healthy trees. And this surprised us really, because we were expecting the smaller trees to have been damaged more. Whereas the larger trees, a lot of them have 0% canopy cover. So a lot of them are dead or nearly dead. Very few of them are still healthy. And so we thought, well, maybe natural selection has already happened with the younger trees, and only trees with the genetics to be more healthy under ash dieback have survived. And that's why so many of these young trees are healthy. And if that is the case, then that would be very encouraging, of course. We then took 600 of the trees, extracted DNA from them, and we sequenced them, whole genome shotgun sequencing to 10x coverage by Novagene. And Kerry Metheringham has been analyzing these data. So it's a huge data set, and we've been chugging away at this for almost a year now. We found actually access to high performance compute has been quite difficult during the pandemic, because so many biologists are analyzing data because they can't get into the lab. So that's held us up a bit. But we are beginning to get some results now. So the first thing that we've done is look at the SNP loci that we found in our previous study, in that Stocks et al. Paper based on the Forest Research mass screening trials. And we used them to estimate how healthy we thought these trees should be in this natural population. And then we compared that to how healthy they actually were. And so far, our results aren't looking particularly great. So here's the adult trees. On the horizontal axis, you can see our genomic estimated breeding value. And then on the vertical axis, the estimated canopy cover from our eyeballing of them in the wood. And this is for 69 adult trees. And this used 50,000 SNP loci that we used before in the previous study. Only 43,681 of them were actually variable within the population. The other 7,000 or so were fixed, and so weren't helpful to us in making these calculations. But as you can see, our correlation is not particularly good at all. There is a very, very slight increase in our genomic estimated breeding value with estimated canopy cover, but it's very, very slight. What is slightly encouraging is that for the trees that still have over 50% canopy cover, so that's trees up here, we do have genomic estimated breeding values that are positive and in the sort of upper 2/3 of our range. So that's good, but as you can see, overall, the correlation isn't particularly great. When we look at the juvenile trees, the results appear to be slightly worse. So here's again our genomic estimated breeding value. Then here's our health scores. So 5, I'll remind you, is a really healthy tree. 1 is a dead tree. And we're not seeing a relationship. We'd like to see a line sort of going up like this, and we're not. So now we're thinking long and hard about why this is. So this is very hot off the press. It might be that we discover that we've made a small mistake somewhere in the code, and actually, we'll correct that within the next few weeks. That may well happen. Mistakes do happen. It might be that the woodland that we're looking at has quite a different genetic background to the trees in the forest research trials. And genomic predictions are well known not to transfer well between different genetic backgrounds. However, the forest research trials were based on trees from all over the UK, and our results did seem to hold up between the different sites. So we think it might not be that. The other thing we have to remember is in the forest research trials, the trees were in very uniform conditions and were all the same age. So the environmental variability was low. However, in the natural woodland, we've got trees of very different sizes, very different ages, and very different microenvironments. They're different distances from other trees. Some of them are in little hollows. Some of them may have, just by chance, be in a place where they get ash dieback worse than trees just a few meters away that are in a slightly different microenvironment. So it might be simply that environmental variables are just overshadowing the genetics here, and we're simply not picking up the signal of the genetics. So we have a lot to think about to understand what's going on. We haven't finished yet, so please don't tweet this. And we've got a lot of work still to do. The other thing we want to do, of course, is look at some of our other hypotheses, and in particular, one thing is to compare the adult from the juvenile trees. Now, here's a very quick comparison, just using PCA of all of the SNPs, and you can see just the first two principal components plotted out here, and you can see the adults in a sort of pinky color and the juveniles in a bluey color, and you can see that largely the range of variation is similar in the adults, but you could conceivably read into that a slight shift. That might partly be because of pollen blowing in from elsewhere, but it might also be due to natural selection happening. So one thing we're planning to do is try to work out the parentage of all of the young trees and the extent to which we can assign them parents within the adult population, and see if we're seeing shifts in allele frequencies among different age cohorts. So we have a lot of work still to do on this data set, but this is where we're up to right now with that data set. Now we'll move on to our work on oak. And we've been working on oak now for a few years, not as long as we've been working on ash. And for this work, we're collaborating a lot with Sandra Denman and Nathan Brown and their group of collaborators, who are mainly working on the problem of acute oak decline, but also on the problem of chronic oak decline. And we have been sequencing trees from these 5 different populations that Sandra, Nathan, and their collaborators have been examining in Sheen, which is in Richmond Park, Hatchlands, which is a National Trust property in Surrey, Chestnuts Wood, Attingham Park. Chestnuts is in the Forest of Dean, Attingham is a parkland, and Langdale is in Malvern, and it's also a very human influenced piece of oak woodland. And we were supplied with samples from these sites by Sandra Denman, Nathan Brown, and the Forest Research Technical Services Unit, and we extracted DNA at Kew. Here's the guys who are involved in that. And we sequenced 436 with Novagene, and we got about 22 times coverage of all of those trees. Here's the health statuses of those trees as recorded by Nathan Brown and colleagues. And 4 of the sites have got acute oak decline, with some trees currently having bleeds and symptoms of AOD, some of them showing signs of being in remission, of having had it in the past and recovering. Some of the trees are healthy. In Attingham, some of the trees were too young to be scored for AOD. And then Chestnuts is actually a chronic oak decline site. The area that we were looking at doesn't have acute oak decline as far as we know. Gabriella Nocci, who's a PhD student with me, funded by DEFRA, and all of this work is funded by DEFRA, has been analysing this data. And this is a PCA plot just based on the 4 sites that have AOD. And this is based on over 2 million unlinked SNPs in 386 oak trees. And the first thing that you can see is Quercus petraea and Quercus roba are separated. Here's a cluster of Quercus petraea, and here's a cluster of Quercus roba. And then we can also pick up the hybrids between them. That's in principal coordinates 1 and 2. And then here's principal coordinates 1 and 3. And again, you can see, perhaps slightly more clearly in this one, the separation between roba and petraea and the hybrids between them. And here's a fast structure plot, another way of looking at this. Here's petraea, here's the hybrids, and here is roba. So there's very good evidence here of hybridization going on between the two species, something that has been talked about a great deal in the literature, and something that can be quite hard to pick up on from morphology. Gabriella looks at the genetic variability in genic regions, the genetic diversity in genic regions and intergenic regions across all of the chromosomes of the oak genome. And we used a genome assembly of Quercus roba made by the French INRA group led by Christophe Plomion that published the genome of oak a couple of years ago. And we used their annotation. And you can see that in intergenic regions, there's higher genetic diversity than in genic regions. And that is, of course, what we would expect to see. In all of these trees, Gabriel has also been able to assemble the whole chloroplasts. And he's put these into a network. And here you can see that network. It's essentially the same in A and B here, but in A, we've colored in the different sites that the trees are from. And in B, we've colored in the different species that the trees have been assigned to. And you can see that many of the chloroplasts are found on many of the different sites, and they're also found across the two species. And this fits well with previous work. Now, there's some huge data sets that were assembled in the past where people looked at chloroplast variation using restriction enzymes. And we wanted to be able to compare our results to these. So Gabriella did some in silico digests of his assembled genomes so that he could come up with the fragment sizes that you would expect to see for them if they were digested with the restriction enzymes used in the previous work. And so that means he's been able to compare our results with results gathered quite a few years ago by people like Joan Cottrell at Forest Research. And they gathered a lot of data from ancient woodlands, whereas we're looking at some highly managed sites. Most of the sites we're looking at are parklands. And so one of our questions was, can we find any evidence about the seed sources for these parklands? Did the owners of the parklands source their seeds locally, or have they brought in acorns from elsewhere, maybe even from different parts of Europe? And what we're finding is that the chloroplasts that we're finding fit quite well with variation found locally in ancient woodlands, according to the data assembled by Joan Cottrell. So we're not finding any evidence that these parklands were planted up with oaks from the continent or derived from elsewhere. We're just starting to enlarge our data set, and we're doing this based on the Forest Research Forest Condition Survey. And this is a really nice study that's been going on for very many years, for decades. And unfortunately, the Forest Research Forest Condition Survey was discontinued a few years ago, but recently funding has been made available for the sites to be revisited and for the oak trees in particular to be scored for their health statuses. And when that new field work was done, leaves were collected by the Forest Research Technical Service Unit and sent to us at Kew on silica gel. And we've been sitting on them for a few months, but we've just got funding from DEFRA to get the DNA extracted and sequence the genomes of a subsample of these trees. So this will give us a really nice UK-wide data set of oak genomic variation, and that will gradually build up our data set of oak. And we're hoping that we'll eventually get to the point where we can start to understand some of the genomic basis of resistance to acute oak decline, and perhaps also chronic oak decline, and also look at genome environment associations to look for local adaptation in oak populations across the UK. And that might help us to suggest where seeds should be sourced for future planting, especially under climate change. So this is very much a work in progress. And I'd like to thank DEFRA for funding essentially all of the work that I've just presented today. And of course, our various collaborators, both on oak and ash, and in particular, the Woodland Trust and Forest Research and Rothamsted. Thank you very much.
Brilliant. Thank you, Richard. Really fascinating talk. For our delegates online here, Richard is one of the pioneers in this area, and it's very nice of you to share a lot of your new data as well, especially showing where quite significant challenges are starting to pop up and things not working quite as you expect. But I mean, it's great for the community to see that and to think about, and for hope